Hello friends, in this module we will learn about the germ theory of diseases. People once believed that diseases are caused due to foul odors or evil spirits or even planetary alignments. The predominant theory until germ theory of diseases was eventually accepted in the 19th century was termed the Miasma Theory, meaning pollution or bad air. Miasma Theory stipulated that disease originated from the decomposition of organic matter, causing a noxious vapor harboring disease-causing agents. It was believed that individuals could contract disease by inhaling foul-smelling air associated with contaminated drinking water, unsanitary conditions, and air pollution. The picture here is a representation of cholera epidemic of 19th century, which depicts the spread of disease in the form of poisonous air. However, with the subsequent development of new experimental work, research, and new theories of different researchers surfacing, the older theories were replaced by the germ theory of diseases, which is currently accepted scientific theory of diseases. Even before microorganisms were seen, some investigators suspected their existence and the responsibility for diseases. Among these, the Roman philosopher Lucretius suggested that diseases are caused by invisible living creatures in his written work. Yet another scientist was the physician Girolamo Fracastoro, who suggested the theory of contagion. He proposed that infectious diseases are caused by minute invisible particles. According to him, infectious diseases spread from person to person by minute invisible seeds or seminaria that are self-replicating and act on the humors of the body, that is blood, yellow bile, black bile and phlegm, to cause disease. His theories were ahead of their time, and it took more than 200 years for the microscope to be invented and his theories to be proved. Anton van Leeuwenhoek was a Dutch cloth merchant and a lens grinder. He had an insatiable curiosity and a desire to do everything for himself. Now, as a tailor, he needed to examine the quality of cloth. So instead of buying a magnifying lens already available, he ground his own lenses to make microscopes. One fine day, he observed a drop of pond water and was astonished to see small moving creatures. He called these small creatures as animal cubes, which meant very small animals. The questions that were asked surrounding Leeuwenhoek's discovery were not related to what the microbes did, but where did they come from? Some people suggested that the microbes caused rotting, while others thought they simply gravitated towards rotting. The third idea was that these microbes generated spontaneously. The first to provide absolute proof for germ theory was Agostino Bassi. He was an Italian entomologist who was the first to show that a microorganism could cause disease. He demonstrated in 1835 that the silkworm disease by the name muscardine was due to a fungal infection. This parasitic fungus is now known as Beauveria bassinia. Not only this, he also suggested that many diseases were due to microbial infections. Alas, there was still much work to be done by the germ theorist in order to finally convince the opposition of its validity. Ignaz Semmelweis was a Hungarian physician working in the Vienna General Hospital. He noticed a striking difference between the hospital's two maternity wards. One ward had an alarmingly higher rate of childbed fever than the other. He thought that the reason for the high rate of childbed fever was due to the ward's student demography. The ward was full of medical students who performed both autopsies and deliveries without much care to clean up. The low rate in the other ward was probably due to the fact that only students of midwifery performed deliveries. This theory was vindicated when a professor died of what was assumed to be childbed fever after cutting his finger in the midst of an autopsy. Semmelweis then suggested and made it compulsory for doctors to wash their hands with a mixture of chlorine and water. Childbed fever death decreased dramatically after the implementation of this practice. Germ theory's wheels of motion began accelerating as it was finally received positively by the medical fraternity that diseases could be transmitted interpersonally. The man who would come to be thought of as the pioneer of the germ theory is none other than Louis Pasteur. He was a professor of chemistry at the University of Lille, France. Pasteur was the most powerful opponent of spontaneous generation theory. He firmly believed that microorganisms were present in the air and were being deposited on all the objects. Winemaking was an important industry in France then. The distillers were worried because the quality of their wine was deteriorating. In the summer of 1856, M. Bigot, father of one of Pasteur's students in chemistry, called upon Pasteur to help him overcome difficulties he was having manufacturing alcohol by fermentation. Pasteur investigated the fermentation process and found that their problems were due to conversion of sugar to lactic acid by rod-shaped bacteria. His microscopic observations made him realize that when alcohol was produced normally, the yeast cells were plump and budding. But when lactic acid would form instead of alcohol, small rod-like microbes were always mixed with the yeast cells. He found that the yeasts characteristic of the alcoholic fermentation had been replaced by bacteria which brought about lactic fermentation which made wines sour. Pasteur then developed a process of heating the grape juice just enough to kill most contaminating bacteria without changing the juice's basic qualities so that it could be then inoculated with yeast to ensure that alcohol fermentation occurred. Today, pasteurization, as this process is called, is routinely used in fermentation and dairy industries. Moving ahead, in the middle of the 19th century, the French silkworm industry was being destroyed by infectious diseases which were killing a great number of silkworms. In 1865, Louis Pasteur accepted the request of the French government to investigate pebrine, a silkworm disease, though he knew nothing about silkworms. For several years, Pasteur struggled but eventually succeeded in isolating the parasite causing the disease. The parasite was found to be a protozoan. Thus, he once again proved that diseases are caused by microorganisms. After several years of research, Pasteur was able to save the silkworm industry by developing a method of preventing contamination of healthy silkworm eggs. The method was soon employed by silk producers all over the world and is still used in silk producing countries. After silk, Pasteur turned to wool and tackled the problem of anthrax, a disease of cattle. The causative agent of anthrax had already been identified by the time Pasteur entered the arena. It was none other than the great German physician scientist Robert Koch who had isolated the anthrax bacillus. However, definitive proof that it was the isolated bacillus and not any other agent that was carried in the original medium which was responsible for causing the disease was still lacking. Pasteur inoculated a drop of infected blood from a sheep drying from anthrax into 50 ml medium and incubated it and repeated the process of dilution 100 times. Thus, the original sample was so highly diluted that there was no chance of any molecule from the original sample remaining in the culture. Yet, the last diluted culture was as active as the first in producing anthrax. As only the bacillus itself, by growing each time in the new culture, could escape dilution, it proved beyond doubt that the anthrax bacillus and nothing else could be responsible for the disease. Pasteur continued to make discoveries concerning the cause and the prevention of infectious diseases. Louis Pasteur conducted numerous experiments in his career to demonstrate the relationship between germs and diseases. He established without doubt that certain diseases are caused by microorganisms. Joseph Lister was a professor of surgery at Glasgow University. He was worried about the fact that many people survived the trauma of an operation but died afterwards due to post-surgical infections known as ward fever. In the Edinburgh hospital where Lister worked, almost half of the surgery patients died from such infections. Lister read about Pasteur's work on how wine went bad because of microorganisms in the air. Lister wondered if the microorganisms in the air were also the cause of the infections which killed his patients after they had successfully survived surgery. Probably the open wounds made it easy for the germs to get into the body. He started to clean the wounds of his patients with carbolic acid and soak the dressings in the antiseptic liquid as well. He invented a machine that misted out portions of carbolic acid into air around the operation table and equipment to kill airborne germs. He then experimented with hand washing, sterilizing instruments, and spraying carbolic acid in the theatre while operating in order to limit the infection. Note the person spraying carbolic acid in the operation theatre during the surgery. In the years from 1864 to 66, the death rate for Lister's surgical patients was 45.7%. Between 1867 to 70, when he introduced this new antiseptic treatment, this fell drastically to 15%. For his invaluable contribution, Louis Pasteur was eager to acknowledge Louis Pasteur. Lister was very eager to acknowledge Louis Pasteur's invaluable contribution. In a letter to Pasteur in the February of 1874, Lister wrote, "Thanks for having, by your brilliant researches, proved to me the truth of the germ theory. You furnished me with the principle upon which alone the antiseptic system can be carried out. Hospitals all over Europe began improving their sanitization by invoking the uses of gapless metal instruments, glass operating tables, increased antisepsis, and disallowing outside clothes in operating rooms. Today, although the antiseptics and disinfectants being used have changed, aseptic surgery is still the basis of saving millions of lives, and Joseph Lister is rightly called the father of antiseptic surgery. Robert Koch was born in 1843 and was a German physician by profession. He is considered to be one of the founders of bacteriology for his various contributions in this field. As the district medical officer, his primary concerns were the prevalent occurrence of anthrax among farm animals in his area. After converting his four-room home into his own medical laboratory, stocked only with a single microscope, Kosh set out to determine the cause of this virulent disease. He was working on anthrax around the same time when Pasteur was working on it in France. Let us now discuss how Koch led us to the germ theory of diseases. Koch established by careful microscopy that the causative agent, a rod-shaped bacteria, was always found in the blood of an animal suffering from anthrax. He took a small amount of infected blood and injected it into a healthy mouse. The healthy mouse showed symptoms and succumbed to the disease. Now, he took a small amount of blood from this first infected mouse and injected it into the second mouse. The second mouse too succumbed to the disease. He could then take the blood from the second mouse, inject it into another and obtain the same characteristic disease symptoms. He repeated this process as many as 20 times and proved that the bacteria did indeed cause anthrax. The 20th mouse died as fast as the first, and in each case, Koch could demonstrate the rod-shaped bacteria by microscopy. This is one of the first original photographs of anthrax bacilli published by Koch. Note the shape of the bacteria. What are they called? Yes, they are called bacilli. Koch carried this experiment further. He found that the bacteria could also be cultivated in the nutrient fluids outside the animal body, and that even after many transfers in culture, the bacteria could still cause the disease when re-inoculated into an animal. Bacteria from the diseased animal and the bacteria in the culture both induced the same disease symptoms on injection. This led him to formulate 4 postulates. On the basis of these and other experiments, Koch formulated what we now call the Koch's postulates for proving that a specific type of microorganism causes a specific disease. Koch's postulates are: the organism must always be present in all infected animals but not in healthy ones. The organism must be isolated from the infected animals and grown in pure cultures. The characteristic symptoms of the disease must be produced when the pure culture of the organism is inoculated into a healthy animal. The 4th postulate states the microorganism must be re-isolated from this experimental animal and cultured again in the laboratory. This organism must be the same as the original microorganism. Using Kosh's postulates as a guide, the investigators following Kosh identified the causative organisms of many human and animal diseases. The greatest accomplishment of Koch in medical bacteriology was his work on tuberculosis. Koch's aim was to demonstrate the causative agent of tuberculosis. He isolated the causative agent Tubercle Bacillus. He realized that the mere identification of the organism associated with tuberculosis was not enough. In order to prove that it was the specific cause of tuberculosis, he needed to culture the organism. The bacteria, however, could not be grown on routine culture media. He succeeded in isolating and growing the organism using coagulated blood agar. Koch announced his discovery of the tubercle bacillus in the famous lecture given in Berlin in 1882. Students, there was a fierce rivalry between France, led by Louis Pasteur, and Germany, led by Robert Koch, to prove their supremacy in the field of science. Hence, at the 10th International Medical Conference in Berlin, Koch felt forced to announce good news that he had found a cure for tuberculosis called tuberculin. Countless patients and doctors traveled to Berlin to obtain the remedy. Tuberculin, however, proved to be ineffective as a therapeutic agent. Koch didn't want to admit his failure and so administered tuberculin to hundreds of people who developed TB and died. As enthusiasm for the putative remedy waned, Koch's reputation declined. In the midst of public scandal, Koch fled Germany for Africa. However, the word of the scandal never reached the entire populace. Koch had many other triumphs in microbiology besides discovery of causative agent of anthrax and tuberculosis. These included the discovery and isolation of the causative agent of cholera, Vibrio cholerae. In 1896, Koch later went to South Africa to study the origin of rinderpest, and although he did not identify the cause of this disease, he succeeded in limiting the outbreak of it by injection of bile taken from gallbladders of infected animals into healthy farm stock. This was followed by his work in India and Africa on malaria, water fever, surra of cattle and horses, and plague, and the publication of his observations on these diseases in 1898. Soon after his return to Germany, he went to Italy and the tropics, where he did useful work on the etiology of different forms of malaria and their control with quinine. Thereafter, Koch continued his experimental work on bacteriology and serology. In 1905, Koch received the Nobel Prize in Physiology and Medicine for his work in tuberculosis. After a roller coaster history, germ theorists finally prevailed in proving to the world that disease is, in fact, often caused by microorganisms multiplying in their host. Thus, germ theory of diseases is that certain diseases are caused by the invasion of the body by microorganisms. This is based on three basic underlying principles. The first, microbes can cause illnesses within the body. Second, microbes and thus illness can spread from one person to another. And third, a specific microbe exists for each illness which will always invoke the same illness. The observations and actions of Ignaz Semmelweis and Joseph Lister would retrospectively be acknowledged as contributing to the acceptance of germ theory. but it was the laboratory researches of Louis Pasteur in the 1860s and then Robert Koch in the following decades that provided the scientific proof for germ theory. Their work opened the door to research into the identification of disease-causing germs and potential life-saving treatments. Today, the germ theory of diseases still remains a guiding theory that underlies contemporary biomedicine.